Hello, Bio 240 students. We are going to start our semester off with introduction to the topic of phylogeny. So this is going to relate to both naming different kinds of living things and also organizing them in a way so that we can make sense of the vast diversity of life. So hopefully you're already familiar with the classification system that we've been using in science for hundreds of years. And we have a way of naming and organizing all different kinds of living things. We started out using categories kingdom, phylum, class, order, family, genus, species. And more recently, we added domain on top of that. So we have 3 domains. Those are the bacteria, the archaea, and the eukarya. So the bacteria and the archaea are kinds of living things that only have prokaryotic kinds of cells, whereas the eukaryotes have eukaryotic kinds of cells. And so the eukaryotes are the only of the 3 domains that have multi-celled life. So bacteria and archaea cannot be multi-celled, they're only single cell. All right, so we have domain as the very highest level of classification. And then once we look inside the domain of the eukaryotes, we find that there's a variety of different kinds of living things that are quite different from one another. So we put them into different kingdoms. For example, we have a kingdom for the animals that's different than one for the plants. Obvious to even small children that animals and plants are different for the most part. There's some tricky ones, but we'll come back to those later on. All right, so kingdom animals. Inside the kingdom of animals, there's many kinds of animals. We're going to divide those into phyla. So on here it says phylum Chordata. When we only have one, we call it a phylum. When we have many, we'll call them phyla. So that's the plural. Chordates have more or less a spinal column or a backbone, internal skeleton. So chordates are going to include not just mammals, but also birds, reptiles, amphibians, fish. But we're going to look at the mammals. Within the mammals, so the mammals is a class within that phylum. Now, phyla are going to be things that are quite different from one another. In fact, by the end of this semester, you're going to be able to tell apart just by eye fairly quickly and easily about 10 different animal phyla. So those things are quite obviously different from one another still, although we still can tell pretty easily that they're animals. So when we look inside the class of the mammals, we see there's a variety of different kinds of mammals. This example of a leopard is Pantheroleo, so it's a carnivore. We can tell a lot of different kinds of mammals by their skulls and teeth. So carnivores are going to be the ones with the big pointy canine teeth, and they eat meat or carne. So when we look at the carnivores, we're going to have cat-like carnivores, the Felidae. We're going to have dog-like carnivores, the Canidae. So those groups within the order are the families, and then within families we have a genus or genera if there's more than one. So the genus is going to be capital P, Panthera. The species is Leo. That identifies uniquely a kind of living thing, a species. So we'll come back to how tricky the species concept is a little bit later, but there's lots and lots of examples of organisms that we can obviously tell apart from one another. A wolf is different from a lion, and so those are going to be things that we have unique kinds of living things that we're going to call species. So that species name is kind of like our name, a first and a last name, except that we also exist within a whole family. We have relatives, we have grandparents, we have cousins, right? So these other levels of classification lets us know what other kinds of species are related to our species of interest, and also what other kinds of species are similar, most similar to it. So where we have other species that are inside the same genus, they're most similar to one another. Then we have other species within the family, but in different genera. Those are going to be less similar to our leopard than they are to the kinds of organisms in the same genus, right? So it's really important to understand this idea of nestedness of the taxonomy. So there's 2 other ways you can think about this system. One is if you have, say, rooms with filing cabinets in them. Each domain gets its own room, and then within the room, there are filing cabinets. Each filing cabinet has drawers, and then inside the drawer, there's green file folders, and then within them, there's manila folders, and within them, there's individual sheets of paper. So these different levels of categories are like those levels of organization that we would have in a filing system. Another way to think of it is like an address. So our physical address, we get down to the equivalent of a species, it would be a house and a number. So whatever street you live on and that number, right? But there might be University Boulevard in many cities and towns across the US. So just giving our address as that street number isn't our full address, right? So we might want to start with saying we live on planet Earth, and we live in the continent of North America, and we live in the country of the United States. And in the United States, we live in the state of California and the county of San Diego. And then if we're proud of our neighborhood, we might say we live in that whatever area you live in. And then, a city of San Diego or a city of El Cajon. And then we can get down to our street number and address, and that will identify our physical location uniquely. So these hierarchies are really important to understand. Right, so this is a system that not only organizes, but also names our organisms. We call that two-word name, the Pantherolio, we call it the scientific name or the Latin binomial. And that's a way that we can always know exactly what organism we're talking about. We may have many common names for organisms. We might call it a leopard. We might call it something else in a different language. We might call it a kitty, whatever we call it. If we use that scientific name, everybody knows exactly we're talking about the same thing. So let's look at this idea in a little more detail with some examples. So again, when we see that we have two kinds of dog-like animals, they're going to be together in the genus Canis. Canis latrans is a coyote, Canis lupus is a wolf. Canis familiaris is going to be our domestic dog, and it's going to be in that same genus too. So they are in the family Canidae, and those are dog-like animals. So notice there's also Phylogeny on this diagram, and that it's only showing one genus and one species, but you know that there's many more kinds of cat-like animals that are being shown on this diagram, so we aren't showing them all. Now we have another family on here, the Mustelidae. These are mammals, they're carnivores, and they're different from cats and dogs because they have musk, scent glands, so they're kind of smelly things. And within that group, we're going to have a genus of Taxidae, which is for badger, and we're going to have Lutra for river otter. So sea otter is also in there. It's in Hydrolutra. So we have a variety, we can have a variety of different species within each genus, many genera within each family. And we see on this way of presenting these animals, there's a relationship between them, and that the branches of the diagram are showing how closely related these different species are to one another. So this is showing that the Mustelidae are more closely related to the Canidae than both are to the Phylogeny. So we're going to look in more detail at this kind of diagram and make sure that you all understand exactly what those parts of the diagram are trying to show. All right, there's a lot on this slide. We're going to stay here for quite a while. So notice that there is a angled line going from lower left to upper right, and that's one part of the branching tree diagram. Then these other branches start to go off to the left, and we see that large arrow on the far left-hand side where it says time. And so that means that the very bottom of the diagram is the oldest in time, and the top of the diagram is the most recent in time. So we're going to use these evolutionary tree diagrams, or cladograms, and we're going to show that species that are still alive today are going to be at the tips of the branches. If we want to show that there's a species that's extinct, then we can show that that branch ends somewhere back in time compared to where we are right now. And so we might have a branch that ends lower down. That would show that that species were extinct. Right, so we have places on this tree diagram where we have lines that intersect one another. So those are called branch points or nodes, N-O-D-E-S. And this diagram is indicating that where we have one of these branch points or nodes, that is representing a speciation event. Now, this is going to be a place in evolutionary history where two different subgroups within one species that was starting out started to separate from one another and diverge in their characteristics. So we're going to have evolution happening that's going to cause changes, or that changes are happening as a result of evolution. And so those forms, the bodies or the DNA sequence or some characteristic of those organisms is going to be changing over time. And it's changing in such a way that it's getting more and more different from the original group they started with. So we'll see that these branch points lead to different species with different characteristics ending up towards the top of the tree. So what those characteristics are and how we can use them to tell these groups apart is very important. So we have these little black bars on this diagram, and if we look here, we see that this black bar is representing what's called an apomorphy. And an apomorphy is going to represent the fact that there was an evolutionary change in a characteristic. On this tree diagram, we're assuming that there is an ancestor that we might find in the fossil record that's down here. And we can know what characteristics it had based on looking at its skeleton or inferring from other evidence that we have. Whatever characteristic that ancestor had, we're going to say that's the ancestral state. And when we have an evolutionary change that happens here, we're changing from that ancestral state to a new state. And so we're going to call that new state a derived state. Right, so the derived state is the new one. And we're going to assume that every species that evolved after this point on the tree diagram is going to still have this characteristic. So as we go up the branches of the tree diagram and we have more evolutionary changes happening, we're seeing the characteristics of each of the taxa at the tips is going to be more and more different than the one that's similar to the ancestor. So this diagram is showing that this species over here, taxon A, doesn't have any changes from the ancestor. So it looks very much like the fossils that we find in this group. An example might be a horseshoe crab or a shark that looks very much like it has for millions of years. Then we start to see changes happening, and off on this branch, these three characteristics changed, and then these two species separated apart from one another here near the top of the tree. So this means that both taxon B and taxon C have all three of these characteristics, right? And they also have this one. Right. Now we see here that there's one characteristic that's only on the branch for taxon C. That's how we're going to be able to tell it apart from taxon B, right? So now these two taxa that are most closely related to one another, we're going to call them sister taxa, just most closely related, shown on our tree diagram. All right. So we have this kind of evolutionary change. We're going to call it an apomorphy. Now, when we have multiple apomorphies, or any apomorphies that are shared by more than one taxa, we're going to say, well, these taxa, B and C, have these shared derived characteristics because they share them together. So we have a special name for that. They're not just any old kind of apomorphies. They are synapomorphies, shared derived characteristics. So using shared derived characteristics to build a tree diagram of the evolutionary relationships between our taxa is really the fundamental task that we're doing when we're using cladistic phylogeny. And that we want to make a distinction between these shared derived characteristics, which are ones we're going to use to tell which organisms are closely related. And then we're going to be able to distinguish between closely related groups because we're going to look for some character that allows us to tell one group from the other. So we're going to look for a unique derived characteristic. Those are going to be called odd apomorphies, like a self characteristic. So we have an odd apomorphy over here for taxon D. Any apomorphy that is only on one of the branches of the tree is going to be an odd apomorphy for that group. It'll be unique. That's how we'll tell that group from the others. So there's a lot of vocabulary here, and it's really important to be able to determine the differences between what these tree diagrams are showing and what these relationships mean. So suppose we looked at taxon E there. Just want to make sure that you think about what are the characteristics that taxon E should have. Well, if we trace its track back here, we know that it has to have this one, and this one as well. And taxon F doesn't have an odd apomorphy, so actually we can't tell the difference between taxon E and F on this diagram. There's one more characteristic or vocabulary word I want to explain while I'm on this diagram. Okay, and that is when we look at any set of taxa, if we're going to group them together and say, okay, here's these two taxa. Suppose these are two different species, and I want to say these are in the same genera as one another. I want to make sure that if I'm going to call this a genus, that I'm including all of the taxa that belong there. And so the way we're going to do that is to look for what are called monophyletic groups. Monophyletic means one lineage. We're going to be able to tell if it's a monophyletic group because if we were to clip any of the branches on the tree, then everything that would fall off if we clipped it would be our monophyletic group. And we have many in the same tree. So we could clip the tree here on this long branch and we get taxon B and C, that's a monophyletic group. If we clip the branch over here where it says synapomorphy for taxa D, E, and F, if we clip the branch off here, we get D, E, and F together, that would be a monophyletic group. And the entire tree diagram taken all together is also a monophyletic group. So we're going to have multiple nested monophyletic groupings, all shown on the same tree. All right. Now, the reason that we might have groupings that are not monophyletic, where we, for some reason, have decided to group together two taxa, where we'd have to clip two different branches in order to put that grouping together, it usually turns out that we've made some kind of mistake, that maybe we have new information or that somebody disagrees with us in how to interpret that information. And this is a diagram that's showing the different subgroups within the phylum of the annelids, the segmented worms. So oligochaetes are segmented worms like earthworms, Hybridinias, leeches. And then we have these marine segmented worms that we used to call polychaetes. All right, so polychaetes, we used to think that all polychaetes were all monophyletic, they all belong together, and then there was this branch over here that the terrestrial annelids all belong to. So in this case, if this were true, then we would have the oligochaetes and Hybridiniae together are monophyletic groups, and then all the polychaetes over here are monophyletic grouping. So a relatively recent large genetic analysis of all of these vast diversity among polychaetes discovered that, in fact, oligochaetes and Hybridiniae are a little subgroup over here, and they're a monophyletic grouping, but that we have multiple side branches off of this tree, and that trying to call polychaetes a taxonomic grouping is not a monophyletic grouping. That the way that they're branch off of the tree diagram is going to show us that what we used to think of as being a unique and complete evolutionary grouping actually is not. So usually we use this term monophyletic versus not monophyletic to reflect that we have had a change in understanding of how our evolutionary groupings work. I'm going to pause right here and I'm going to plug in my computer. Right, so when we're talking about traits that we're using to build our evolutionary tree diagram, it turns out that some characteristics can be tricky, that we might see two different taxa that both have a characteristic, but they each evolved it by themselves, separately or independently from one another. So we can have two different things be true. When we look at these two different tree diagrams, the one on the left shows that we have two taxa and they both have a derived characteristic, but their most recent ancestor also had it. So that evolutionary change occurred for this monophyletic grouping before they diverged into different species. And we're going to call that a homologous characteristic. So these two species have this characteristic because they had a shared ancestor that already had it. So we can also see species that both have a characteristic, but their last most recent shared ancestor didn't have it. So that tells us that each of these different species, again, showing on the black dots at the tip of the branches on the right-hand tree, that each of those species evolved that trait anew by themselves. So evolution had to happen twice of this trait. This has happened quite a few times in evolutionary history in a variety of different taxonomic groups. For example, birds and mammals both have a four-chambered heart, but it evolved in each group separately. So the shared ancestor of both birds and mammals was a reptile that had a three-chambered heart, and then mammals diverged from reptiles and separately evolved a four-chambered heart, whereas birds are derived from different reptile ancestor and they evolved a four-chambered heart as well. Now these structures of the four-chambered hearts are slightly different. If we look at them through development, we see that they're slightly different four-chambered hearts, but if we just count up the chambers, we would say that they both had a four-chambered heart. All right, so we need to look out for what's called convergent evolution, and that is that wildly different taxonomic groups can sometimes have characteristics that look the same, but they both evolved them because they allow the species to survive and reproduce better. So shared environments often lead to convergent evolution. So when we look at these characteristics, that's a more historical and traditional way of looking at how similar species are to one another. Since the 1950s and 60s, we have been able to start using technology where we look at DNA sequence. So when we talk about DNA sequence data, we're going to say, okay, this is molecular data. This is different from using characteristics of the skeleton or behaviors or anything that isn't proteins or DNA. When we look at different types of evidence, we see that we can look at proteins that are going to be a way to allow us to distinguish between different genetic changes. We used to be able only to look not at the DNA sequence, but proteins that were made from DNA sequence. And we call these allozymes. So when we found a different, slightly different protein, we could infer that different genetic sequence made it. And blood types are an example of a protein that can be different between different groups based on evolution. We might be able to look at types of DNA that exist only in one copy in the genome. And so these are called haplotypes rather than genotypes. And we find those looking at mitochondria, chloroplast DNA, or looking at the Y chromosome. When we look at nuclear DNA, the DNA that is in the nucleus, there's two kinds. There's coding and there's non-coding. So coding is going to make a specific protein, and often we'll see a paper that's using DNA sequence data, and maybe it's looking at a ribosomal protein, or it's looking at some kind of DNA sequence for certain enzyme. And when we look at non-coding regions, we find that there's a lot of diversity between individuals. These non-coding regions can mutate and change much more rapidly than coding regions, because if a gene changes enough, it isn't going to work. And so we see a lot more flexibility of changes. So when we look at these different kinds of non-coding DNA, these are the ones we're going to use that allow us to do these fingerprinting techniques, where we might be able to identify an individual. So different kinds of DNA technology that we can use, we might call them microsatellites, or tandem repeats, small tandem repeats, STRs. Mini satellites are very small tandem repeats. We might also have a paper we're reading where we see the acronyms RAPID, RFLPs, AFLPs, or SNPs, SNPs. So these are all different kinds of DNA that we're looking at. And when we look at these nuclear non-coding DNA, we're going to be looking for relationships between very close recent evolutionary history. When we look at coding regions, usually we look at those mitochondrial genomes. We're looking for much more ancient changes. So looking for major differences between taxonomic groups. So depending on the type of DNA sequence we're looking at, we can ask evolutionary questions that go very far back in time, or that are happening much more recently. And so we're not going to get into any of the details, but when you start to look at papers, research papers that are using different types of molecular data, we may see that different techniques, different genes that are chosen, may give us slightly different versions of proposed evolutionary tree diagrams. So although they're very powerful techniques, they can also result in there being multiple possible trees that fit the evidence. All right, so here's an example of what DNA sequence is going to look like when we're trying to use it for phylogeny. The way that we're going to use it to say we don't have different character states like 4-chambered heart or 3-chambered heart. We're actually going to look at the letters in the genome and see if we have the same letter in the DNA sequence. So if you look at the fly eyeless gene and we look at the shark and you look across and you see, oh, well, they both start with GT, but then there's an A and for fly and there's a G for shark. Those are differences. So across the very top, you're going to see the DNA sequence for mouse. We look down, we're going to see which letters are the same. And you can kind of squint in this and see that the more letters that are the same, you're going to see sort of more green, and the more letters that are different, you're going to see more white, right? And then these proportions are summed up. So we look at fly, its genetic similarity to mouse is 76%, whereas squid, it's 78%. Right, so we're just going to use these percents to say how genetically similar. So on these, among these choices, shark with 85% is going to be the most genetically similar to mouse. And we're going to interpret that, right? This is a big tricky thing. How do we use DNA sequence data to propose how closely related species are? Well, the more percent similarity, the more closely related they are. So we're going to say shark and mouse are most closely related. If you look carefully at this slide, you'll notice, however, even though there's DNA sequence data differences, the protein that this gene makes comes out identical, right? Because we have those codons that code for amino acids, and we can have different codons that still code for the same amino acid, so we can still get exactly the same protein. And so if we were trying to look at this gene just using allozymes, which only look at proteins, we would say, oh, they're all the same. We can't tell. But if we look at DNA sequence data, we can see these very, very fine scale differences. All right, so when we look at a cladogram, before we've just had them sort of floating in the air, but here we have put an x-axis across the bottom of the diagram. And where we have fossil data, we know when a certain species diverged from another one. We can put a specific x-axis in years going across the timeline. So we can say that all of the bears that are living today diverged apart from one another really more recently than 10 million years ago. So they're relatively closely related compared to how long ago the bear lineage split off from the raccoon lineage. When we look at DNA sequence data from much more recently, we can also use what's called the molecular clock. So we have an estimate of how fast mutations happen, so we can calibrate how different groups are apart from one another just based on seeing how many mutations different they are. All right, we just have a couple more things to talk about as far as tree diagrams before you're going to be ready to start working on your own. This is a tree diagram showing all the different species of Galapagos finch, and it's a little more complicated diagram. And we can see that there's a lot of different branching points, and that these finches are grouped together sort of by the habitat they live in and by the foods that they eat. And we see that their beak size and shapes are very different from one another. Something that's important to keep in mind about these tree diagrams is that there's some things that we can change about them that don't change the way they show evolutionary relationships. There's other things we change about them that do change the way they show evolutionary relationships, right? So imagine if instead of a piece of paper, that this tree diagram were a mobile, which is a kind of movable sculpture or art or toy. Sometimes you put these over a baby's crib. So a mobile is going to be something, maybe it's got a string dangling and it has a twig, and then it has hanging from the twig, it has the little thing. So if we turn this upside down, we'd have the common ancestor dangling there, and we'd have these other branches hanging. And they can turn. And if they twist, they're actually showing the same evolutionary relationship. Let me show you a couple ways that we can show trees that show the same relationship. We can draw them so that their root is to the left. We can draw them so that their root is down. And then we have either the angled kind of V-shaped branch points, or we have the square or football goalpost shaped branching points. And these tree diagrams all show the exact same relationship between these different species. It's just convenience or your personal preference of how you want to draw them or how it best fits onto your paper. So it's a good idea to try practicing drawing the trees these different ways and see which one you prefer, which one makes sense to you, and which one is easiest for you to draw, because there's no right or wrong here. You can do it anyway. This figure is showing the idea of this rotation. So any of the vertical twigs on our tree, we can rotate it, right? We can turn it like so, and we're showing the same relationship. So here we have, look at where the taxa X is. If we rotate around that branch, we can put X on the other side, and we're showing Z and Y there. But where they split away from each other is still the same. It's still showing that W is closest to the ancestor, X is next most closest to the ancestor, and then Z and Y are sister taxa that diverged most recently, and that are the most different from the ancestor. So look carefully at those tree diagrams that are across the bottom of the slide. Make sure that you understand that they are all showing the same evolutionary pattern. All right, so when we start the activities for this lab, there's two components. There's one that's drawing trees by hand, and we'll get into that in a minute. The other one is you're going to use some actual DNA sequence data, real DNA sequence data from a series of published papers that are investigating the relationship between bats and other mammals. So bats come in sort of two kinds. There's megabats, or the larger kinds, the fruit-eating bats, and they're larger and sort of fuzzier and cuter looking. And then there's microbats, or the insectivorous bats, and they are going to be the ones that are flying around at night eating bugs. So the question these scientists were looking at is, are megabats and microbats sister taxa? Did flight evolve once in mammals so that all bats are closely related? Or do the megabats, who have those cute little furry faces, are they more closely related to primates, which are similar in many ways? So some primitive primates are bush babies or flying lemur. And then are the microbats, which eat insects, are they more closely related to tree shrews? Shrews are a small mammal that are kind of like mouse-sized, but they're carnivores, they eat insects. So the question that the scientists were trying to answer using some DNA sequence data was, what are the relationships between these different taxa? So you're going to go through the instructions in a PDF file attached to the lab that's going to allow you to actually use real tools that scientists use every day in order to analyze DNA sequence data that they have. So this is one of the wonderful things about modern science, is that everybody who comes up with some DNA sequence, they are going to load it up into this giant database called GenBank. And then anybody else who wants to use it for their research is going to be able to do that too. And then also there are free analysis tools that different scientists have built and published to do the computer intensive and complicated steps of actually analyzing those DNA sequence. Because there's thousands and thousands of letters, not just 6 or 8 like the characteristics we're using. So you're going to do that activity, and you're going to figure out the answer to it. And then we're also going to work on some paper and pencil. So if we want to try and draw a cladogram, we're going to need to have our taxa and our outgroups. We're going to need to decide if characteristics are ancestral or derived. And then all we have to do is tally up those characters, and we're going to use the number of differences from the ancestor to construct our tree. And we're going to, sometimes we're going to end up with more than one tree that could be possible. If there's a difference in the number of evolutionary steps that it takes to do these different trees and make them work out logically, we're going to assume that the one with the fewest evolutionary changes is more likely. And that principle is called parsimony. We want to choose the most parsimonious tree. That is the one where the fewest evolutionary changes had to happen in order for it to be drawn in a logically correct way. All right, so get your lab manual out, and then you can open up the other file, which is the instructions for working on a practice cladogram.